Hey guys, welcome to this quick part one of two-part video on gene expression and regulation. Part one here is meant to give you a quick boost and kick off into gene regulation, what it is, what you should be thinking about. So you had an assignment where you're starting to think about protein synthesis and some of the big components of that as a bit of review. And then we'll move into the regulation of that protein synthesis. So here's something that we know about DNA. DNA is going to code ultimately for proteins. Those genes specifically are going to code for proteins. And inside of all of our cells, we find a nucleus if we're eukaryotic, like we are, and there's DNA inside of those eukaryotes, inside of every single one of our cells. Gametes have half the amount of DNA. Things like red blood cells don't have a nucleus, so there are exceptions to this rule, but inside of most of our cells, we have a nucleus that contains the exact same set of DNA. That's sort of the big message, is every cell has the same DNA. If that's the case, if all cells have the same DNA, then how do we end up with all these different types of cells? And we know that these differentiate. So here are some examples of a cheek cell, or down here we've got some nerve cells, or we've got some red blood cells over here with white blood cells. We've got all these different types of cells that make up our very complex bodies and body systems and tissues. If every cell has that same DNA, then how we end up with different types? And that's really due to gene expression and regulation, certain genes being switched on and off. So the easiest way to regulate genes is by regulating their transcription, or the first part of protein synthesis, where RNA polymerase is going to make a copy of a gene of interest. There's two ways we can regulate that. There can be on and off switches in which genes are turned on, actually allowing DNA to go into RNA, or those can be shut off and they stop transcription. There's also dimmer switches. And they're going to interfere with the transcription process, so they can actually slow it down, which means you slow down the production ultimately of proteins. So here are the two big systems that we'll look at. First is prokaryotes. These are simpler. Eukaryotes, like us, are much more complex in how we regulate transcription, how we regulate genes. This is going to regulate at prokaryotes level. They're going to regulate at the level of transcription. And the two examples that we need to know, these are the two that we're really going to focus on, are the lac and trip operons, right? So you have an assignment today to take a look at the lac and the trip operon and begin seeing how these things work. Eukaryotes, they'll regulate at almost any level. Differential gene expression is going to lead to cell differentiation eventually. This is much more complex, and we're going to go over this in a lot of detail with each other. But prokaryotes, they are going to regulate at the level of transcription, and we need to know about the lac and the trip operon. So here's an example to sort of kick you off. You have a scenario where you have some E. coli, and E. coli is a type of bacteria, Escherichia E. coli, that lives in your colon. What it needs is an amino acid called tryptophan in order to survive. Let's say that you aren't eating enough tryptophan-rich food in order for that amino acid to get into the E. coli. So what the E. coli is actually going to do is recognize that, and it's going to make its own. So it's going to make a protein. It's going to switch on a gene that makes a protein that creates it. If you do happen to eat tryptophan-rich food, well, then the E. coli doesn't need to switch on that gene that makes the protein that will create it. And the evolutionary reason for that is it's going to save energy. Why have a gene switched on and making something if you're getting it already from your environment? That doesn't make any sense. So that will get switched off. That's an example of what we mean by gene regulation. If there's a lot of something present, then maybe something gets switched off, and we don't necessarily need to make a protein that's giving us something that we need to make that's already coming in from our environment. Right, so that's what prokaryotes do. They switch genes on and off depending on what's present in their environment. Okay, so if E. coli can turn on genes, or they can make the tryptophan, and they can turn off genes because tryptophan is already available, how do they do that? They cannot simply recognize that there is tryptophan present or there isn't tryptophan present using a nervous system. So how do they do this? And that's where we get into gene regulation and something called operons. And remember, these operons are just in prokaryotic systems like Escherichia coli. So that's where we're going to start. These have to do with feedback loops, different types. The trip and the lac operons are different types of operons, but they give you a good example of how these systems work, and those are the two that we really need to know. So that's a quick primer for what gene regulation is. I'll be back tomorrow to talk to you guys about this idea in some more detail.